Look, up in the sky. What do you see? No, not the bell tower. Not McMurrin either. It's all around us, and even makes up the air we breathe. We are not talking about oxygen, though. We are talking about nitrogen. Nitrogen makes up about 78% of the air present in Earth's atmosphere, making it the largest component of air taken in by living organisms. However, nitrogen in the atmosphere does not exist in a usable form by organisms. Oh, look over there. What is that enzyme under the tree doing? Based off of his name tag, it must be nitrogenase. It appears that he's working really hard. Nitrogenase is an enzyme that facilitates the process of nitrogen fixation, therefore providing chemical energy to living organisms and the biosphere. Let's take a closer look at how it works.
Nitrogenase is synthesized by nitrogen-fixing bacteria. Some of these bacteria exist independently, such as cyanobacteria, also known as blue-green algae. Other nitrogen-fixing bacteria form symbiotic relationships with plants. These bacteria live in specialized nodules on the plant roots and help to provide the plant with necessary nitrogen.
Nitrogenase has been extensively studied and is therefore largely understood. Nitrogenase is composed of two subunits, a tetramer molybdenum iron complex and a dimer iron protein. The iron protein contains the sites for ATP binding and hydrolysis, whereas the site for substrate binding and reduction is located on the molybdenum iron protein. The iron protein breaks down ATP to generate electrons in the form of ADP, and these electrons are then pumped into the molybdenum iron protein in order to drive the conversion of nitrogen. The molybdenum iron protein is composed of two unique metallosulfur clusters, the P cluster and the homocitrate iron molybdenum cluster. This iron molybdenum cluster is bound to the molybdenum iron protein through both a cysteine sulfur and a histidine ligand, and is believed to be the enzyme substrate binding and reducing site, also known as the active site of the enzyme.
The overall reaction of nitrogenase involves 8 hydrogen atoms, 8 electrons, and N2, or dinitrogen, which is found naturally in the atmosphere. The overall reaction serves to convert dinitrogen into NH3, or ammonia, which can be more readily utilized by the plant as a source of energy. This reaction additionally produces excess hydrogen. The initial step in this reaction takes place in the first subunit of nitrogenase, which contains an iron atom and serves as a reductase protein. In this reaction, electrons are pumped from the iron subunit into the molybdenum iron protein, which contains the active site for nitrogenase. The 8 electrons are pumped from one subunit to the next, and this energy is provided by the hydrolysis of ATP into ADP. The second subunit, which contains the molybdenum iron cofactor, is the active site of nitrogenase and is the point where the broken down dinitrogen is fully converted into ammonia. The active site of nitrogenase is an iron molybdenum cofactor held in place by a histidine and a cysteine residue. As electrons travel from the P cluster to this cofactor, the transferred energy is used to break the bonds of dinitrogen and form ammonia. The exact mechanism by which the bonds are broken and new bonds formed between nitrogen and hydrogen is not yet fully understood.
The nitrogen cycle starts with N2, or atmospheric nitrogen. This is what most of the air we breathe is made of. However, it cannot be utilized by plants or animals for metabolic purposes. This is where nitrogenase comes into play. Atmospheric nitrogen is diffused into the soil where our friend nitrogenase can absorb it. It is found in the nodules of plants and fixes nitrogen into usable ammonium, or NH4+. Other bacteria found in soil can then take ammonia and convert it into nitrite, NO2-, or nitrate, NO3-. Atmospheric nitrogen makes its way back into the air through a process called denitrification. Nitrate, nitrite, and ammonium can be taken up by roots of plants to be used in metabolic processes. When plants die, this nitrogen is taken up by decomposers, like mushrooms, or it can be ingested by animals, like this little bunny. When either the mushroom or bunny die, their nitrogen can be taken up by soil bacteria, and the cycle begins again. Nitrogenase is so important because of its ability to fix atmospheric nitrogen into usable nitrogen for plants and animals. The synthetic creation of nitrogenase is energetically expensive, and to avoid this, plants take advantage of a symbiotic relationship with bacteria to convert nitrogen for them. Living organisms are then able to use this nitrogen to build proteins and nucleic acids.
So in conclusion, nitrogenase is a vital aspect to the nitrogen cycle and allows for unusable nitrogen in the air and soil to be converted to usable ammonia for chemical energy. So thank you, nitrogenase, for living a life of significance and allowing us to build the next great University of America. Continue doing what you do and set the world on fire.